Okay, welcome back to the CTSI Distinguished Lecture Series at UCLA. It's my honor to introduce Marco Colonna, who is the Robert Roch Belliveau Endowed Professor in Pathology and Immunology and Professor of Medicine at the Washington University School of Medicine. Marco is a member of the American Society of Clinical Investigators and also was elected to the National Academy of Science. Marco's career has been filled with many trailblazing discoveries, including discovering the role of human plasmacytoid dendritic cells during human influenza infection, the cloning of leukocyte Ig-like receptors and TREM receptors, and identification of inhibitory natural killer cell receptors. He's also discovered various subsets of innate lymphoid cells in mice and humans and profiled their roles in homeostasis and disease. And more recently, Marco's group has demonstrated that the loss of TREM2 and human variants commonly associated with Alzheimer's disease impact the ability of microglia in the brain to respond to beta amyloid plaque deposition. So Marco's group has since continued its interest in neuroimmunology with a recently published study in Science, discovering a lymphopoietic niche at CNS borders, which he is going to highlight today. So Marco, thank you for being with us here today. Looking forward to your talk.
All right. Well, thank you, Tim, for inviting me. It's a great pleasure to be, although virtually, at UCLA. And of course, thank you for this kind introduction. Let me just share the screen now. Oops. Okay. There you go. Share the screen. And okay, this should be it.
Yeah, that looks good.
Okay, good. So what I'd like to do today is to tell you our recent work in which we look at lymphopoiesis in the central nervous system. And what we know, of course, for a long time that lymphopoiesis occur in the bone marrow. But what I'll show you today is that the central nervous system, brain and spinal cord, have a private source of lymphopoietic hematopoiesis. And perhaps we speculate, we envision that this source of lymphopoiesis is important to maintain tolerance in the central nervous system. So the work I'm going to present today was done by a very talented postdoc in my lab, Simone Brioschi, together with another postdoc, Weili Wang, and a graduate student, Vincent Peng. So let me remind you that the brain, the central nervous system, particularly brain, is enveloped by three different membranes, the meninges, which insulate the brain for the rest of the body. The outermost membrane is the dura mater, which separates the brain from the periosteum and the bone. The intermediate layer is the arachnoid, which sends this delicate trabeculae to the connective tissue to the pia mater, and the pia mater then separates the meninges from the glial limiting membrane and the brain parenchyma. Now, the space between the arachnoid and the pia mater is called subarachnoid space. This space contains the cerebrospinal fluid and also rich networks of blood vessels that traverse the meninges and go into the brain. So we know for some time now that the meninges, the dura, and the arachnoid, the pia mater, contains immune cells, both innate and adaptive. Neutrophils of particular abundance, which you can also find monocytes, macrophages, NK cells, ILCs, and also adaptive immune cells like T cells and B cells. Now we notice that the meninges contain a very enriched in neutrophils and also contains a large percentage of B cells. Approximately 30% of the cells in the meninges, of immune cells in the meninges, are B cells. Presumably, all these cells come from the circulation. So the first question we ask is, where are the cells located in the meninges? Are they located within the meningeal tissues, or they're located in the blood vessels that traverse the meninges and go into the brain? And so to address this question, we took CD19 tomato B-cell reporter mice and then inject them with tomato-lectin 488 to visualize blood vessels. And then we performed 2-photon imaging on a cranial window. What you can see here, highlighted in green, are the blood vessels in the meninges. And in red, you can see the CD19 positive B cells. And you can see that some of B cells are found within the blood vessels, but the vast majority of B cells are actually in the extravascular space. In this little movie, you can see again these B cells moving slowly in the meninges, and then you can see B cells moving fast with the blood flow into the vessels. The second observation we made is that the B cells that we find in meninges contain at least two subsets, immature B cells. We look in the literature at meningeal B cells that have been previously profiled by single-cell RNA-seq, by Ido Amit, and by Burkhard Becker. And we found that in both studies, B cells include two subsets of cells that were defined as mature B cells and immature B cells. And we reclustered actually the data from Ido Amit's data. This is the B cell cluster, and then we look at differential expression markers. All B cells express CD19 and express C79B or IG beta, but one subcluster of B cells expresses markers of mature B cells like MHC class 2 or MS4A1, and another cluster or subcluster of these B cells expresses markers of mature B cells like RAG1 and CD93. Now we know from studies on B cell development and maturation that mature B cells express a high level of membrane IgD and low levels of membrane IgM, whereas immature B cells and transitional B cells express high level of membrane IgM and low level of membrane IgD. So we look at the expression of IgM and IgD in B cells from different compartments. Now, in the blood and in the spleen, the majority of B cells are all mature, and so they express high level of IgD and intermediate level of IgM. But when we look in the dura, we see that a small percentage of cells express high level of IgD. Some B cells express high level of IgM and low level of IgD. We have transitional B cells that express both IgM and IgD. And also we have double negative cells that express no IgD and IgM, which presumably represent pre-B cells. And interestingly, we find the same profile in the dura and in the bone marrow. Now, the other important information that we know from studies on B cell development is that B cells need to rearrange their immunoglobulin gene through activation of RAG, and therefore RAG is expressed during development, but it's not expressed in mature B cells. And so we look at the expression of RAG in the meninges by using RAG1 GFP reporter mice. And so here we look particularly at the top of the meninges, top down, and we focus on a sagittal sinus, which is particularly rich in immune cells. And then we look at the expression LIFE1, which indicates vessels in the meninges, IgM, which indicates immature and mature B cells, and RAG1 GFP. And you can see that in the meninges along the sagittal sinus, you see many cells express RAG, which presumably are pre-B cells, and also cells that express both RAG and IgM, which represent cells that are maturing and becoming mature B cells. We also know from studies in B cell development, of course, that RAG1 is important for the generation of mature B cells, and indeed, rag knockout mice have no mature B cells in the blood. They do have some pre-B cells in the bone marrow, and we found also the same pre-B cells also in the dura. And finally, we performed single-cell RNA-seq of CD45 positive cells in different compartments, in the dura, in the blood, in the bone marrow. And then we focus our attention on the clusters of B cells in these 3 different compartments. And so when we look in blood, we see that the vast majority of the B cell clusters that we identified express markers of mature B cells, and they poorly express markers of mature B cells. When we look in the bone marrow, we see clusters that express markers of mature B cells, but we also see clusters that do not express mature B cell marker, but express immature B cell marker. Again, RAG1, CD93, Alzheimer's receptor, and also MKI67, which is a marker of proliferation. And in the dura, we see a similar trend. We see cluster of cells that express mature B cell markers and cluster of cells that express markers of immature B cells. Finally, we recluster all the single-cell RNA-seq data into one single UMAP, and then we projected the B cells from different compartments onto this UMAP. And you can see that blood B cells occupy one territory of this UMAP, whereas the bone marrow and the dura together occupy similar territories in this UMAP, which is quite distinct from the territory in the blood. And we further look at the composition of different clusters. And so when we look clusters of mature B cells, we see that cells here are contributed mostly by blood B cells. When we look at pre-B cells, we see that these are contributed mostly by bone marrow and the dura. When we look at maturing B cells, we see these are contributed both by blood and by bone marrow and the dura. When we look at B1 B cells, they're mostly contributed by blood. Proliferating cells are mostly contributed by bone marrow and the dura. And finally, pro-B cells derive almost exclusively from the bone marrow and the dura. And finally, we confirm the presence of pre-B cells and immature B cells by confocal microscopy. Here we look at the meninges, we stain them with different staining. These B cells are always from the CD19 tomato reporter mice. We stain them with KI67. We see that some B cells are actually proliferating. We see some B cells express CD93, a marker of mature B cells. And when we look at CD20, which is a marker of mature B cells, some of B cells express CD20, but some of B cells do not express CD20, suggesting that these are immature or pre-B cells. So this is an interesting observation because we know that blood does not contain pre-B cells and contains very few mature B cells, which seem to be abundant in the meninge. And so we ask the question, where do meningeal B cells come from? And one obvious answer to this question was the skull bone marrow for several reasons. First of all, we know the skull bone marrow, particularly the top of the skull bone marrow, the calvaria, is rich in bone marrow. We find bone marrow in the transverse sinus, and also we find bone marrow along the sagittal sinus. And by histology, you can see abundant bone marrow in the calvaria. We also have isolated B cells. We found abundance of immature B cells that express high level of Alzheimer's receptor, and we also find hematopoietic stem cells. But the other reason why we thought about the skull bone marrow is because previous studies, at least three different previous studies, have shown that there's a direct connection between the skull and the meninges, particularly dura mater. 3 studies have shown that there's some blood vessels that can go from the skull into the dura, and they have to go through these ossified vascular channels. And so this ossified vascular channel provide a direct connection between the bone marrow of the skull and the dura mater. And in this image, you can actually see better these vascular channels that connect the bone marrow of the skull up here with dura mater down here. So our hypothesis is that perhaps B cells, particularly mature B cells and pre-B cells that we find in the meninges, derive from the skull bone marrow. And so we, to test this hypothesis, we performed this skull bone marrow chimera. In this experiment, we irradiate the head of the mice, but we protect the rest of the body with a lead shield. And then we transplant them with bone marrow from a congenic strain, 45.1. And then after 4 weeks, we look at B cells in different compartments, in a tibial bone marrow, in the blood, in a skull bone marrow, and in the dura. And we see whether the B cells come from the host or the donor. In a tibial bone marrow in the blood, we see the vast majority of B cells come from a host, not surprisingly, because we didn't irradiate the body of a mouse. But when we look in a skull bone marrow, we see that a very large percentage of the B cells actually come from the donor. And we see the same percentage of B cells come from the donor also in the dura, suggesting that indeed B cells in the dura derive from a skull bone marrow. We also perform some experiment of confocal microscopy using CD19 tomato reporter mice, which were injected with tomatolactin, again, to visualize the vascular connection. And we look at the expression of CD19 positive B cells and also their expression of IgM. Again, you can see these vessels here, these vascular channels, which connect the bone marrow with the dura. And you can see B cells going from the bone marrow into the dura. And some of these B cells are IgM negative, so presumably pre-B cells. These are additional images. Again, we visualize these channels connected to skull bone marrow with dura. We see a number of cells of CD19 positive B cells in these channels, and we see that only a few of these cells express IgM. So we wanted to, furthermore, we want to validate even more the migration of B cells from the bone marrow, from skull bone marrow into the dura. And so we performed Parabiosis experiment, in which, in this Parabiosis experiment, we take wild-type mice and CD19 tomato reporter mice. We join them together through the skin to allow circulation of blood cells from one mouse to another. We do the Parabiosis for 4 weeks to let B cells equilibrate, and then we look in a wild-type parabion how many cells of CD19 tomato parabion we can find. And so in the blood and in the spleen, we found about 40 to 50% of the B cells derived from the parabion, suggesting that there's free movement of B cells from in the two mice, between the two mice in the blood and the spleen. But when you look in the brain or in the dura, we see that a small percentage of B cells derive from parabion, suggesting that the vast majority of B cells in the brain and in the dura are actually derived from the host parabion. We also performed another experiment in which we took wild-type mice and inject them with CD19 tomato splenocytes, and then look at the donor B cells in the mice after 1 week. And so in the blood, in the spleen, we see a significant percentage of B cells derived from the donor, but in the brain and in the dura, we see very few cells derived from the donor. Again, suggesting that the endogenous B cells in the meninges are not derived from the circulation. So the next thing we ask is, what drives the migration of skull B cell progenitor into the meninges? And to address this question, we first look at the expression of receptors and ligands that are known to mediate migration of B cells. And so we look here at the expression of a number of different molecules in B cells, and we also look at the expression of their ligands in fibroblasts. And we notice that B cells, particularly immature B cells like mitotic B cells, pre-B cell, pro-B cells, express high levels of CXCL4. And the meningeal fibroblasts, on the other hand, express very high levels of the ligand CXCL12. In addition, we use this algorithm, this niche-net analysis algorithm, which can predict the interaction between receptor and ligands based on the level of expression of different receptor-ligand pairs in different tissues. And so, again, when we compare this level of expression of B cell receptor and the level of expression of their ligands in fibroblasts, we see once again the CXCL4, CXCL12 interaction emerges as one of the most significant interactions. We also noticed a second important interaction between TACI and APRO. This interaction is known to be important to mediate survival and development of B cells. We wanted to validate the relevance of interaction between CXCL12 and CXCL4. And to do this, we took reporter mice for CXCL12 and then injected with ometolectin to visualize blood vessels. And then we look at the distribution of CXCL12. And as you can see from this imaging, we find that in the meninges, there's a very extensive production of expression of CXCL12 in the meningeal fibroblast along the blood vessels. In addition, we have isolated these fibroblasts from the meninges, stain them with CD31 and podoplanine, and then look at the expression of CXCL12 in different subsets. And we found that this particular subset of fibroblasts expressing both podoplanine and CD31 is highly enriched in the expression of the CXCL12. We also look at the expression of CXCL4 in different subsets of B cells in the bone marrow and in the dura. Not surprisingly, we find that the expression of CXCL4 is inversely correlate with the maturity of B cells. So if we divide B cells into early, late, and mature B cells, we see a progressive reduction of the level of CXCL4 expression. So together, these experiments suggest that the migration of pre-B cells in mature B cells from the skull bone marrow into the dura is driven by CXCL12, CXCL4 interaction. Now, we also ask whether meningeal B cells can exit the brain. How do they exit the brain and where do they go? And we were helping this by our collaborator, Yoni Kipnis, who has discovered a few years ago that the meninges, particularly dura, contain lymphatic vessels. These lymphatic vessels drain the cerebrospinal fluid. They drain macromolecules, but they also drain cells, and they drive macromolecule and cells through lymphatics into draining lymph nodes, particularly deep cervical lymph nodes. And so we wanted to know whether B cells can also go through these lymphatic vessels, exit the meninges, and reach the deep cervical lymph nodes. And so to address this, first of all, we performed some confocal imaging. We took mice. We look at the meninges, particularly, again, the sagittal sinus and transverse sinus, inject the mice with ometolectin to visualize a blood vessel, and then stain them with LIVE-1, which detects lymphatic vessels, and B220, which detects B cells. And so you can see here both blood vessels and lymphatic vessels along the transverse sinus and sagittal sinus. And you can see that within the lymphatic vessel, there's a number of B cells that are clearly found within the lumen of the lymphatic vessel. To confirm that the cells can migrate through the lymphatic vessel, we took CD19 tomato splenocytes and inject them directly in a cisterna magna, which has access to the cerebrospinal fluid. And then 1 hour, 24 hours after injection, we look at the presence of injected B cells in the deep cervical lymph nodes or in irrelevant lymph nodes like inguinal lymph nodes. As you can see, almost none of these cells can reach the inguinal lymph nodes, but we see a significant percentage of donor B cells in the cervical lymph nodes. And finally, we look where these cells go. So once you can see, once again here, the B cells, they go through the lymphatics, and once they reach the deep cervical lymph node, they're mostly found in the B cell area of the lymph nodes. So what I've shown so far is that the mouse meninges contain B cell progenitor, immature B cells. The B cell progenitor derived from the skull bone marrow. That the migration of B cell progenitor is likely driven by CXCL12, CXCL4 interaction, and that B cells exit the meningeal compartment and through the lymphatics and can traffic to the draining cervical lymph nodes. Now, we know that B cells that develop in meninges are exposed to CNS antigen. So we ask the question whether this can facilitate immune tolerance against CNS antigen. And so to address this question, we did a couple of experiments. In the first experiment, we took these mice transgenic for a B cell receptor specific for myelin oligodendrocyte glycoprotein. We isolated B cells from these mice and then look at the percentage of B cells that are specific for myelin oligodendrocyte protein by labeling MOG with biotin, and then look by flow cytometry and the percentage of antigen-specific B cells. We look at antigen-specific B cells in two different compartments, in the tibial bone marrow and in the dura. So when we look in the tibial bone marrow, we found that about 20% of B cells are specific for MOG. But when we look in the dura, we find that only 10% of the cells are specific for MOG, suggesting that approximately 50% of the B cells have been negative selected. Now, MOG, of course, is the antigen only expressed in the central nervous system and not expressed in the periphery. We wanted to validate this observation in a different system. And so we use another BCR transgenic, a BCR transgenic for a nominal antigen called HeNa glycozyme, or HEL. We isolate the B cells from these mice and look at the percentage of B cells specific for HEL by flow cytometry by staining B cells with HEL conjugated with Alexa. Now, HEL, of course, is not expressed in the mouse, and so to express HEL in the central nervous system, we inject in HEL directly in the cisterna magna, such that HEL can access directly the cerebrospinal fluid. Alternatively, we inject it with very irrelevant antigen, OVA. And so when we inject mice in a cisterna magna with OVA, we see that 90% of B cells in a tibial bone marrow are specific for maintain their specificity for HEL. And in the dura, we see approximately 90% of the cell maintaining their specificity for HEL. But when we inject the mice with HEL in the central nervous system, we see that in the tibial bone marrow, 90% of the cells maintain their specificity for HEL. But in the dura, in the central nervous system, we see that only 50% of the cells have maintained their specificity. So again, we have lost, probably for negative selection, about 50% of the cells to maintain a tolerance to this CNS antigen. So this experiment suggests that exposure of developing B cells to CNS antigen facilitate immune tolerance against CNS antigen and preserve the immune privilege within the CNS. Now, the data I've shown you so far, our data were obtained in young adult mice, but we're also interested to see what happens during aging. And to address this question, we performed single-cell RNA-seq of B cells isolated from young mice 12-week-old mice, or all mice, 25-months-old mice. And we compare the B cell subset in these two at these two different ages. So we found a number of different B cell clusters. Most of them are common between young and age, but we saw two important differences. The first difference is that age mice exhibit this additional subset of B cells called ABC, which is not present in young mice. These ABC cells are called age-associated B cells. They've been described a long time ago, and I'll talk more about these B cells. We also noticed that plasma cells in age mice are expanded compared to young mice, and this is actually quantification. You can see that age mice have a very large representation of ABC and also very large representation of plasma cells. So age-associated B cells, or ABC, have been reported for some time now. They were initially discovered by Pepper Merrick, and they've been extensively studied by other B cell biologists, for example, by Mike Cancro. We know that they derive from follicular B cells. They're stimulated by CD40 ligation, CPG, and they also require for the development interleukin-21 and interferon gamma. And upon development, these associated B cells express TBET, and the expression of TBET drive their particular phenotype, particularly the expression of C11C, C11B, and other myeloid markers. And that's one of the reasons why these B cells very often are co-purified with microglia, because they both express CD45 and they both express C11B and C11C. However, the cells are B cells, they're CD19 positive. They also express a high level IRF5, and they've been implicated in autoimmunity. In fact, they produce autoantibodies, and they are particularly abundant in mouse models of lupus, in mouse model autoimmunity, and they're also they have higher frequency in autoimmune diseases in human. In addition, these ABC are particularly frequent in female and women, and for this reason, they've also been associated with increased frequency of autoimmune diseases in women. So we look at the phenotype of the cells, in particular genes that are differentially expressed in ABC versus mature B cells. We found a number of markers that have been previously identified, but we also found some additional markers that were particularly interesting. For example, we found that these B cells, age-associated B cells, express high levels of ApoE, which is a molecule that has been implicated in the pathogenesis of Alzheimer's disease. We also noticed that these cells express very high levels of SYK, which is a marker of activation. And they also express this transcription factor, ZBTB20, which may be important for their development. Now, we also look at plasma cells. Here you can see some images of plasma cells in the meninges by confocal microscopy. They're particularly abundant in aged mice, and they have a typical morphology with a large cytosol, abundant endoplasmic reticulum, as compared with mature B cells or naive B cells. We made some interesting observations also about plasma cells. One interesting observation is the isotype they produce. When we look at young mice, we see the vast majority of the plasma cells in the meninges produce IgA. And this is consistent with a paper that was recently published by Dorian McGovern and Mena Clatworthy that shows that the IgA plasma cells are actually educated in the gut, and they can travel from the gut to the meninges and provide defense with IgA-mediated defense there. But when we look at aged mice, we found that only few plasma cells actually produce IgA. The vast majority produce IgM, suggesting that these plasma cells have a different origin. Here again, we look at plasma cells by confocal microscopy. In young mice, first of all, we see very few plasma cells labeled with CD138 whereas in aged mice, we see much more abundant plasma cells. And then when we look at the isotype that they produce, we see that in young mice, indeed, plasma cells mostly produce IgA, some IgG, some IgM. But in the aged mice, we see very few IgA positive plasma cells. Vast majority of plasma cells produce IgG, and particularly IgM are very abundant. Again, suggesting that the origin of plasma cells in aged mice and young mice are different. And so the question we ask is, where do ABC and where do plasma cells come from? And so to address this question, we pair single-cell RNA-seq with BCR sequencing. And this was worked with in collaboration with Meng Wang and Steve Kleinstein at Yale University. So what we do here is that, first of all, we know that the diversity of B cells is due to the VDJ recombination, and which leads to clonal expression of diverse B cell receptors, and also to somatic mutation. And so what we do here is we isolate CD45 positive cells from the meninges, perform single cell RNA-seq, and in addition to that, we also amplify the VDJ region and perform sequencing of VDJ region through bar tag oligos, such that we can identify for each B cell and for each B cell type also the B cell receptor. And then we compare the B cell receptor repertoire of blood B cells with the Dura B cells. So here, for example, we take mature B cells in young mice. We sequence the BCR from 273 cells and in the Dura, and we find that none of these BCRs can be found in blood, so there's no sharing. If we look at aged mice and we sequence 378 BCR from mature B cells, and we find that only a very small percentage of these BCR can be found also in blood. But when we look at the repertoire of ABC in the Dura, and we sequence 247 cells, we see that about 30% of this BCR can also be found in ABC from the blood, suggesting that most likely the ABC in the meninges derived from the ABC in the blood. We did the same type of analysis for plasma cells. In aged mice, we sequenced approximately 130 BCRs, and we found that very few of these BCRs are also present in the blood. But when we look at the same BCRs and we look at the presence of these BCRs in the ABC, we found that up to 15% of these BCRs are present in the ABC, suggesting that plasma cells may actually derive from the from ABC. So what I've shown you is that during aging, ABC, based on our BCR sequence analysis, may derive from the blood and then accumulate in the meninges. Now, we speculate that because ABC have developed in the periphery and not in the CNS, they're now educated to CNS antigen, and therefore, once they reach the meninges and once they reach the antigen, they become exposed to CNS antigen and may differentiate into CNS reactive plasma cells. So this is to summarize what I've shown you. I've shown you that the skull, and particularly the calvaria, provide a B cell, a niche for a B cell lymphopoiesis for the central nervous system. In young adult mice, pre-B cells migrate through these ossified vascular channels from the skull bone marrow into the meninges. They are exposed to CNS antigens at this level, and with mechanism that we're still investigating, B cells are negative selected or edited, and this leads to the generation of tolerogenic B cells for CNS antigen. However, over time, age-associated B cells that develop in the periphery can reach the meninges, extravasate into the meninges, and generate the compartments of ABC which have not been exposed to CNS antigen, and upon encountering CNS antigen, they become activated, and eventually they can generate plasma cells that produce antibodies that could be potentially dangerous because they could lead to autoimmunity. Now, we published these studies already some time ago, and then after our studies were published, 2 additional studies were published that support our studies. In one study, pre-B cells, immature B cells were found in the meninges in steady state by single-cell RNA-seq. And in another studies, again, these pre-B cells were found in the meninges, and it was actually shown with a similar BCR transgenic that we use, the MOG BCR transgenic, that indeed B cells undergo negative selection in the central nervous system. The other point I'd like to make is that this scalp marrow provides a source not only of B cells, but actually provides a source also of myeloid cells. And this is work that was done by Yoni Kipnis in collaboration with us, and that was published together with our paper. They have shown that the scalp marrow can directly supply granulocytes and monocytes to the meninges. And of course, this mechanism provides a first source of the sentinels, myeloid cells in the brain, which can provide a first type of response against perturbation like viral infection or other damage situation. So of course, there's a number of questions that are still open. One question we're interested in is to understand whether meningeal B-cell education is relevant to autoimmune diseases. We're thinking about multiple sclerosis, but also diseases like systemic lupus erythematosus have been associated with neurological manifestation like neurolupus. And perhaps the lack of B-cell tolerance for CNS antigen may be an important mechanism for neurolupus. We're also interested in defining the specificity of meningeal B cells. And the other interesting observation we made is that B cells can travel from the central nervous system into the cervical lymph nodes. So we're interested to know whether meningeal B cells can capture CNS antigen and function as antigen-presenting cells in the draining lymph node, and perhaps modify the T-cell receptor repertoire for and imprint the T-cell receptor repertoire in the cervical node for CNS antigen. So I think that's what I wanted to tell you today. I like to make some acknowledgement. These additional members of my lab that contribute to this work. I'd like to acknowledge Yoni Kipnis, who of course is an expert of meningeal lymphatics, has been instrumental for this work. I'd like to acknowledge Max Artimo's lab. Max is bioinformatician who helped us with the single cell analysis that I present you. These additional collaborators that provide us with transgenic lines and other reagents. I'd like particularly to mention Meng Wan and Steve Kleinstein at Yale University, who helped us with single cell RNA-seq. And also, I'd like to acknowledge my long-term collaboration with Ido Amit at Weizmann Institute for single cell RNA-seq of cells in the central nervous system. So I think I will stop here, and I think we have time to take some questions. I thank you for your attention, and happy to answer any question. Thank you.
Thank you for that fantastic seminar, Marco. So I'll be reading some questions in chat, but while we're waiting for those to populate as well, I know we have one question. I can ask you a question directly to get things started. So I'm curious, like what the air expressing cells are in the meninges, and whether you think that there's some role for ILC3s perhaps in the meninges and antigen presentation to B cells for selection.
So I'm sorry, I didn't get you. You're asking about ILCs.
So first, what are there air expressing cells in the meninges?
Oh, air expressing cells.
Yeah.
We didn't see expression of air by single cell RNA-seq, but of course we have to investigate a little. As you know, single cell RNA-seq does not give you a very deep sequence information, right? So to address that, we should perform more single RNA-seq analysis. But one thing I like to mention is that air is mostly important for selection of T cells, right? And the selection, we have no evidence for selection of T cells in the meninges or in a scalable marrow. In fact, I didn't show you the data. This was an important control in a scalable marrow that we did. In a scalable marrow, I show you that in a scalable marrow chimeras, I show you that a significant percentage of B cells come from scalable marrow, but none of the T cells come from scalable marrow. All the T cells come from the host. And so I doubted that you may have selection of T cells at the level of meninges. You may have selection of T cells at levels of deep cervical lymph nodes. That's a possibility.
Okay, thank you. So Sammy is asking what role do B cells have in meningioma?
Good question. We haven't looked. So we actually look at, we don't have a model of meningioma. And if you guys have a model of meningioma, we'd be happy to know about it. We've looked at human meningioma. We haven't found any similarity with the mouse situation. I guess it's the tumor situation is quite different. What I can tell you is that we look at dura in normal individuals, and we find presence of B cells in the dura, including mature B cells. The problem with human, as you can imagine, is that we have only limited access to the dura. The vast majority of the samples that we have come from diseased individuals, especially for neurodegeneration, and therefore there's a lot of variability between samples, and we're still trying to understand exactly what is the typology of B cells in the dura of human, and this will require some extensive studies.
Okay, thank you. Robert Motlin is asking whether the repertoire of the B cells in the meninges provides an advantage for the response to infection in the CNS.
So first of all, hi, I want to say hi to Robert. It's nice to chat with you on this talk. We're performing experiment in infection, but we don't have any data yet. We're looking at infection by LCMV, intracranial injection, and we will be looking at other infection. We're particularly interested, based on the recent report that multiple sclerosis is epidemiologically related to EBV infection. We're interested in looking at gamma herpes virus 68 infection in mouse. Gamma herpes virus 68 is a mouse model of EBV, although it does not entirely recapitulate the EBV infection. But those are the two types of infection that we're presently looking at. And I think will be interesting to see what happens. I have to say that this is a little complicated because in steady state, the vast majority of B cells are resident B cells. They come from scalable marrow. But I suspect that upon infection and inflammation, you open up the blood-brain barrier, so you'll have both B cells that are derived from a scalable marrow, as well as B cells that come from the periphery. And so we'll have to see what is the relative contribution of these different compartments of B cells.
Okay, Ye Zhang is saying fantastic work. Are meningeal B cells present in developing brains? And if so, how are they different from adult meningeal B cells?
It's a very good question. We haven't looked into that. I have to say that the presence of B cells has been noticed also by Beth Stevens group. They presented some data. They haven't published a paper yet, but what they speculated is that maybe the B cells in the meninge actually come from a separate niche of B-cell development that derives maybe from a Yolk sac or from a very early site of hematopoiesis. However, we're not sure that that's the case because, first of all, we don't see the whole hematopoiesis in the meninges. We see mostly B cells and neutrophils and partly monocytes. And the other reason for that is that we clearly demonstrated that this B cell progenitor come from the skull through the ossified vascular channels. But of course, it's possible that you have during development already you have, upon formation of the skull, you have some migration of B cells from the skull bone marrow into the meninges already during the development. But we really haven't tested that yet.
Okay, if anyone has any other questions, feel free to type them into chat. We still have a few minutes left on the seminar. Or perhaps you can raise your hand and you can address it live to Marco. We'll give a few seconds for that. And if no other questions, then I'd like to thank our speaker, Marco, for being with us today. Thank you for that wonderful presentation, and hopefully we'll see you soon.
Thank you for having me.
Thanks.
Thank you.